you so much for joining us today. Before we kick this off, I do want to point out to the audience that we welcome questions. Please use the Q&A section at the bottom of your screen. Type in questions during today's presentation, and we will address those at the end. So it's my pleasure to turn this over to Steven Saltzstein, Force Family Office's CEO.
So I just want to thank Andrew Schiermeyer, CEO of AvenCell, for joining us today. I mean, what their company is doing in regards to, enabling CAR T, and going after, some of the most difficult cancers in the world is incredible. Their team is world class. And what I love about it is not only are they out there really helping people and curing people, but they're doing so in a manner that greatly increases the size of the marketplace. And so the TAM and really all the potential dollars and revenue that can be generated from this is extraordinary. So here to tell you more, it's my great pleasure to welcome Andrew Schiermeyer. Thank you, Andrew.
Well, thank you, Steve. That was a very kind intro, and thanks, Callie, and the whole Force Family Office for arranging the webinar today. Really appreciate all of you who are joining to listen. I'm Andrew Schiermeyer, the CEO of AvenCell. We started the company in 2021, and joining me today on the phone and ready to answer all of your questions are Olivier Brandicourt, our chairman, who is the former CEO of Sanofi and also Bayer Healthcare, amongst a very prestigious career. Manfred Klevesath, our Chief Medical Officer, Renaud Lescarbot, our Head of Global Research, and Marcus Orchowski, our VP of Finance and Operations. So in a nutshell, executive summary, most of you who are probably on the phone are aware there's a global oncology therapeutics market that's massive. It's over 200 billion, and it's growing at a very high rate of speed. And of this, blood cancers represent around 20 to 25% of it, or a total market of about 50 or 60 billion. Now, many of you have probably have also heard in the past few years, since 2017, there's been a new therapeutic modality called CAR T. It was introduced, and it uses a patient's own immune cells to fight cancer. And since its first market approval, the first 6 products have been approved in blood cancers, but they represent only sort of a modest 6 billion in peak sales compared to the total opportunity in blood cancers of about 50 to 60. And it's estimated that only between 5 and 20% of patients who are eligible for CAR T therapy actually receive it. So we'll talk a little bit about why that is and what the solution is. What's important is that AvenCell, from the start, was created to solve this issue. And the issue is really around scale and profitability of the therapies. So AvenCell has 2 proprietary, highly de-risked CRISPR-based platform therapies that we use, platform technologies that we use. We already have 2 product candidates in clinical studies. These are set to displace current competition within a very short period of time. And the NPV of those 2 alone is conservatively estimated at over $9 billion. Our investment to date includes blue chip investors such as Blackstone, Novo Holdings, and F Prime, and they've collectively added $220 million to date. We also received significant non-dilutive financing from the government of Japan of up to $40 million. And the purpose of the call, and to introduce you all to us, is to raise $30 to $35 million that will generate important value inflection points within the next 12 months that should be able to catalyze an IPO or lucrative industry deals. So before we get to all that, we need to start. The story actually starts in 2012 with a little girl named Emily Whitehead. And at age 6, Emily had acute lymphoblastic leukemia that had already relapsed. And at the time, she was out of therapeutic options and her prognosis was terminal. Emily was the very first patient to join a clinical trial that used an experimental treatment using her own immune cells re-engineered to attack her cancer. And at the time, it was called CAR T, and nobody had heard of it. Today, as I speak, Emily is 13 years later, still cancer-free, and at this point considered cancer cured. So how did that happen, and what is CAR T therapy? Well, CAR T therapy is very basically using your own immune system to attack cancer cells. And your immune system is very complex and has many different kinds of cells. But most importantly, we'll be talking today about T cells, cells that are developed in your thymus. And T cells, if you remember nothing else, they have four very important characteristics that make them really suitable for this fight. Number one, they're endowed with innate receptors that recognize abnormalities on the surface of other cells. The second is that they're actually armed with little, almost hand grenades, if you will, microscopic hand grenades that get unleashed when the T cell binds to another cell that it recognizes as a threat and is able to kill that cell. The third aspect is that, like pretty much every nucleated cell in your body, T cells have a set of self-recognition markers. And that's important because if you want to use a T cell that comes from somebody else, you need to take these self-recognition receptors into account. And finally, T cells have this unique ability that when they do bind to another cell, they reproduce in a clonal way to create daughters and daughters of daughters and so forth in an exponential fashion that allows many clonal T cells to go and attack the cells that they're interested in. Now, for the most part, your T cells are most interested in infectious disease. And when you or I have a cold or a flu, it means that we've acquired a virus that's going around and replicating in our cells. And luckily, our cells, our healthy cells in our body, if they do get invaded by a virus, have a method of showing pieces of that virus on their surface, which is essentially a flag to your immune system that says, "Help, I've been invaded, please kill me." And that's actually represented here by the little red and blue lollipops on the side. So this is where your typical T cell jumps into action. Your T cells have many different receptors, but a particular T cell with a particular receptor will recognize the cell that's been infected, bind to that cell, then kill it, and very importantly, it'll replicate the T cells, which will then go find other infected cells and kill those too. And that's typically how we recover from a typical cold or a flu. Your T cells get involved and they kill all the invading and malformed cells. Now, what's really important, of course, is that the T cell is able to expand massively. And so one T cell that makes that synapse with another infected cell will actually produce in the billions of additional cells and allow you to clear that infection. So where this comes and where this intersects with cancer is that about 20 years ago, some really intelligent people realized that cancer cells could be seen very similarly to infected cells. The difference being that a cancer cell has mutated DNA inside that causes those cells to divide out of control. But nevertheless, a cancer cell will put up flags on the outside that says, "Help me, I'm infected," or something's wrong with me, please attack and kill me. So your typical T cells aren't generally engineered to do that, but now through gene editing and techniques of genomic medicine, we're able to insert a new receptor into an otherwise normal T cell that is geared directly towards attacking cancer cells. And once we do that, the T cell behaves exactly as I showed you before, can attack and kill the cancer cells, can replicate in massive numbers to go out and kill all the other cancer cells as well. And that's where CAR T therapy really gets its origin from. So for a typical patient who has a certain kind of cancer, and in most cases, CAR Ts are approved for blood cancers, they'll go to the hospital, they'll get their blood drawn, the T cells will be isolated from the blood. They'll then go to a central manufacturing site where they're re-engineered into CAR T cells. Those are grown up and expanded into millions and hundreds of millions of cells, shipped back to the hospital, infused into the patient, and they'll go to work against the cancer cells. And it turns out about 50% of patients that can be treated with CAR T achieve a functional cure, and many more of those patients actually do benefit. So this is a phenomenal discovery and a phenomenal development in medicine in the past 10 years. It's helped now thousands of patients who are now cancer-free as a result, and has been just one of the most exciting advances in cancer medicine in the last decade. Now, the question then is, why isn't everybody getting it? And you can see there have been 6 new approved CAR T medicines in the last 8 years. The growth of those over time is pictured on the right, and they're up to about $6 billion in total global revenue at this point. Now, ordinarily, if everybody were getting CAR Ts, this number would be many times higher than this, but that's not the case. And the question is why? So if we look as a case example at B-cell lymphomas, and many of you probably have known someone who have had lymphoma, non-Hodgkin's lymphoma is one type of this, for example. There's 28,000 new cases per year in the US alone. Now, of those patients, of course, when they're first diagnosed, many of them, and even 60% of them will receive chemotherapy, and chemo actually works pretty well in the first line. It renders about 60% of these patients cancer-free for a while, puts them into remission. The other 40% become eligible for CAR T, and of that 40%, again, only 5 to 20% are receiving it. And the question is why? And that is because, as you can imagine, as you saw from the previous slide, CAR T is really tedious. It's very expensive. It can only be done in very sophisticated centers. Many patients who actually try to do CAR T don't have T cells that are good enough to make the product, and many patients actually die while waiting for their CAR T to come back. And as a result, there's a huge opportunity gap here. We know we have a very effective potential therapy, but only a handful of people are getting it. So to make matters worse, it's also terrible for the companies that are making it because the margins are horrible. CAR T is very expensive to make, and if you look at just a case example of two of the bigger approved products for multiple myeloma in an analysis done by Leerink, it shows that the actual cost of goods for CAR T is often somewhere in the $300,000 to $400,000 per patient, meaning that for a lot of these companies, every patient they treat, they're losing money. So there's a lack of scalability, there's an expense and profitability problem for something that otherwise works beautifully and can offer life to people. So what's the solution? Well, turns out the solution is actually quite simple. If we can use donor cells to provide CAR Ts to patients, we can solve pretty much every one of these problems. First of all, scalability and supply. We can get 100 patient doses per donor, and that can be done at any time, completely disconnected from when the patient needs it. So we can stockpile this in the thousands, make it available on demand for the patient. It can be frozen down, no problem. In terms of cost of profitability, because of that scalability, the COGS can be 95% cheaper than autologous at under $10,000 per patient, which renders more than a 90% plus gross margin. And finally, because these are coming from healthy donors, we can always have high, consistent quality because they're healthy. We know the T cells are healthy and we can screen them ahead of time. So that seems like a pretty simple solution, right? We just get donors to give their T cells. We use the T cells for the patients. So why hasn't it happened yet? Well, it turns out that the problem of giving donor cells to patients is a little bit trickier than anybody first imagined. First of all, we can't match the donors to the patients like we do solid organ transplants or even bone marrow transplants. There are just too many people with cancer, and finding perfect matches for those patients is pretty much impossible. The donor cells can be made just like the patient's T cells. They can be made into CARs and they can work perfectly just like CARs, and they can bind to the patient's cancer cells with no problem. But the problem arises if that patient is unmatched to the donor, then the patient's own T cells will go and attack the donor CAR Ts and render them useless. They will eliminate all the donor CAR Ts, and that is exactly what happens. So that's not an option. And over the last 8 years, there have been many companies that have been scratching their heads trying to figure out how can we give unrelated donor T cells to a patient? And that is exactly the solution that makes AvenCell valuable. There's been many tries. Some of them involve eliminating through gene editing all of those self markers on a T cell, but it turns out that your immune system is way too complex and it's able to detect that, and it still rejects those. So that's not an option. So what AvenCell does is we have a 3 sequential CRISPR-based edit step and 1 gene insertion. We knock out what we call the endogenous T cell receptor from a healthy donor's T cell, so that doesn't interfere with anything. We insert the brand new receptor that's geared to go after the patient's cancer, as you saw in the previous slides. And now when it comes to the self-recognition markers, this is the secret sauce, if you will. This is the complex part that nobody else has done, but that we've perfected. We do one gene edit that knocks out what we call class 2 of the markers. And then we do a second gene edit that knocks out not all, but some, all but 2 of the remaining variability of these self-recognition markers. And we leave 2 in. Those last 2 are then matched to the patient in a perfect way so that the patient's T cells don't reject them. And since there are self-recognition markers on the cell, the other components of a patient's immune system also don't reject them. So it's basically a Goldilocks problem where we leave enough recognition on the cell that the patient's T cells and other immune cells are happy with it. And this is what gives AvenCell its value. No other company does this, and we're already in the clinic with it. So with that, let me introduce to you our clinical candidates. We have 2 currently at play. One of them we call AVC201. This is directed at acute myeloid leukemia. And again, this is considered probably the most aggressive blood cancer out there with the poorest prognoses. There are no other CAR competitors of note for us as we speak today. And so we're kind of all alone in this position. No other CARs have been approved for this. We would be the first. The second is a very different profile. It's called AVC203, and this one is aiming straight at the heart of the existing marketed CAR-Ts for B-cell lymphomas and ultimately autoimmune disease. And I'll describe what makes this special in a minute. So when it comes to 201, again, as I mentioned, this is geared towards acute myeloid leukemia, or AML. We do something a little bit more sophisticated in the meantime as well. We have a 2-component system for this one. Instead of binding the CAR directly to the cancer antigen of interest, we have this intermediate step where we have bispecific antibodies that link the CAR to the AML blast. And the reason why we do that is because that enhances safety immeasurably. We are able then to actually control the CAR-T's activity after it's been dosed. If we infuse the linker, it will link up and kill AML blasts. If we stop the infusion of the linker, the CAR-T stops its activity. And this is important just from a safety perspective because patients can develop tolerability issues and severe toxicities because AML is such a tricky thing to do. Nevertheless, we're in dose escalation right now. We've dosed a total of 13 patients. 9 of them are evaluable, and they're shown here. This is a very standard chart in oncology that shows best response of their blast count in percentage. So unlike most graphs, negative here is good. It means they had a reduction by that percent in their blasts. Positive is not good. It means their blasts actually increased. And so from left to right, we're going from lowest to higher doses. What you can see is in the lower doses, we had several patients that progressed. But in the last 4 out of 4 patients that we've treated, we've had very meaningful elimination of leukemic blasts. 2 of those patients had all of their measurable leukemia blasts eliminated entirely, and 2 of the patients had them partially at about 40 and 75% reduced. So we do believe this is very promising. We're on the right track. And one other element that I mentioned earlier is that the T cell expansion is a very important part of its ability to control the cancer. So what I wanted to highlight here without getting too technical is the current marketed CAR-Ts, Kymriah, Yescarta, Breyanzi, those are their trademark names, all expand within the first 9 days in a patient's circulation to levels of around 10,000 CAR-Ts per milliliter of blood. Or there's other metrics of it, but let's just, for the sake of argument, it's around 10,000 CAR-T cells per milliliter of blood, more or less. What happened in the last 8 years is several companies that were trying to do allogeneic or donor-based cells did run clinical trials, but their expansion was far less. And this is a log graph, so it was 10 times lower on average than the cells that come from the patient, the autologous cells. By contrast, this is where we're at already, still in dose escalation. And the mean of our 2.4 milligram day per dose is already consistently 10 times or more higher than existing autologous cells. And in the best case, it's 600 times improvement in expansion over previous allogeneic CAR-Ts. So to the extent that cell expansion is the strongest measure we know of prognosis that the CAR Ts are working and efficacious, this is phenomenal data. It's seriously knocking it out of the park, and even we were surprised to see that it was this good. So the next steps for this program are to finish dose escalation, which we'll do just in the next month or two. We have 4 more patients coming in rapid succession. And if the data looks good, we'll then move to what we call dose expansion, which is another 20 patients done all at the recommended dose, probably at the highest dose that we're doing dose escalation at. And if all that looks good, we'll be done with that by the end of 26, and we'll be into the potential for a registrational trial of around 80 patients. And again, since we're the only ones out there and we don't have anything to compare to, a single-arm registrational trial should be plenty sufficient and very efficient way of going forward. I will remind you that BD has been very active and strategics in the space. Big Pharma has been very active in doing deals for CAR T that they think is promising. And so we have multiple shots along the way at each kind of data readout to think about doing a BD deal that could be very lucrative to the company. Our own estimation in terms of forecast is an MPV, a very conservative MPV of about $5.5 billion at a 12% discount factor. Again, we believe that that's highly conservative, but just gives you a metric that everybody kind of understands. So moving on from 201 to our second candidate, which we call 203. As I said before, this one is quite different. This one now is similar to previous marketed CARs that are already out there on the market, but there are 2 major enhancements. The first is that we're not only targeting one antigen called CD19, we're actually targeting 2, CD19 and 20, and we also have a universal receptor built in that allows us for additional future expansion. That CD19/20 has been designed rationally, knowing that there are new generation CARs coming in the same space, so it's been designed to compete with those. But the second and important aspect of this is it's also allogeneic, and it's the only allogeneic competitor out there that's doing both CD19 and 20. I don't really have the time to go into all the preclinical data, but rest assured that we have done very extensive de-risking, both in vitro and in vivo, with several different mouse models. In our hands, this works as well or better than everything else out there. Our AVC203 suppresses tumor all the way down, even with multiple rechallenges of B-cell tumor models in mice. And it's consistent and similar to the newest generation CD19/20s that are coming, that are all autologous. It's worth noting that it's significantly better than the first generation CARs that are currently out there that aren't able to control tumor in mice at this dose. We've also received phenomenal support from a very wide variety of key opinion leaders. These are all physicians who are top of their field, everywhere from the US to Great Britain to Germany to Japan. Many of these are part of our scientific advisory board. We meet with them all regularly. They consistently tell us that the world needs an allogeneic CD19/20, and that they are very excited to be part of a clinical trial that we're initiating. So many of these are already part of the investigators in our clinical trial. And we're initiating this globally from the start. So we already have EU approval and US approval for this, and we're able to start dosing patients imminently. We're just waiting for our application in the UK, and Japan will follow next year. You may be asking, why Japan? Well, the government of Japan has very generously awarded AvenCell as the first Western-based company they've ever awarded this grant to, $40 million in matching funds in a 2-to-1 ratio over every single dollar of investment dollar from AvenCell to move this program forward. This is non-dilutive financing. There are no hooks into it. The only condition being that we include Japan in the clinical development, which we're more than happy to do. So the program for this, just beginning dose escalation. We intend to finish dose escalation next 12 patients within the next 6 months. That will provide us a very nice data set already to compare to existing CARs and potentially prove ourselves better. And then we'll be in a position to do dose expansion at a recommended dose over the following 12 months, and finally slide directly into a registrational trial immediately after that, which we do assume will be a single arm enrollment and be able to do very efficiently as well. Again, multiple opportunities to do BD deals along the way. As far as we see it, once we're able to demonstrate that this is as good or better than the existing marketed CAR-Ts, several of the large pharma companies who are very vested in CD19 B-cell lymphoma, such as Novartis, BMS, Kite Gilead, AstraZeneca, J&J, they're all aware of this. They're all looking for the data, and we expect that we'll have a lot of interest by the third quarter of 2026. Our expectation in terms of NPV here is about 3.8 billion. Again, that's extraordinarily conservative, and that only includes the very first indication and approvals that we might get. We expect that this has the opportunity to go to 10 billion or more in terms of sales. Wrapping it up in terms of financial and just putting the Gantt charts together simultaneously, what we're solving for here, as I already showed you, AVC 201 and 203, this window in green from the first quarter 2026 through the end of 2026 will be an extraordinarily important one for turning over a lot of cards in terms of efficacy for those two programs. It is within a very short 12-month window where we'll be collecting clinical data that could be very substantial. And what we're looking to do is raise an additional $30 to $35 million for this period, which again would be leveraged by the AMED Japan money to do just that. And at the end of 2026, we'll have the opportunity either to effectuate a BD deal, potentially an acquisition, or continue on with a Series C, or potentially IPO the company, depending on what the window looks like at that time. Just to highlight the fact that the field has been extraordinarily busy over the past year with some very important active BD activity and acquisition activity. You can see them there. They range anywhere from 350 million upfront to 2.1 billion upfront. All of these are in essentially the same areas that we're targeting right now, using slightly different modalities, but also including autoimmune, which I don't have time to talk about today, but also adds a tremendous amount of potential value to AVC 203. So just wrapping up, executive summary here. Hope I've convinced you that CAR-T has been extraordinarily transformational over the past several years, but in its current form, meaning taken from the patient, put back into the patient, it will never be widely available or achieve its full potential. AvenCell is the first company to introduce a CAR-T that's derived from healthy donors that has already shown in the clinic potential to be as good or even better than autologous CAR-Ts. We have two candidates that are prime right now that are either in the clinic or just starting the clinic, so there's not a long wait for clinical data, multi-billion-dollar NPV potential. And we're seeking an incremental investment of 30 to 35 million in a Series C to bring us through the end of 26 and turn over several cards. That money will be directly enhanced in a 2-to-1 ratio for the proceeds on 203 by the non-dilutive grant from Japan. And we do expect continued support from our existing investors, Blackstone, Novo Holdings, F Prime, and others. So just as a final note, our mission and our vision is to give hope to many more patients just like Emily to have a full and a long life, and we're working very hard to do just that. So at that point, let me turn it back over to Steve and Callie and perhaps take some questions if the audience has them.
Great. Thank you so much, Andrew. And you can stop sharing your screen and we'll get into the questions. So if the solution is as simple and inexpensive as you suggest, could this become a frontline treatment for hematological cancers?
Yeah, it absolutely can. And we're already starting to see elements of that. In oncology, what we have to do, based on, ethical concerns, is we always have to start with patients who have no other options. So we always start at the very back of the line with the sickest patients. And as we demonstrate safety and efficacy, the standard is to move up through those lines. So we've already seen over the past few years CAR T as a modality moved from fourth line to third line to second line. And now, in some cases, it is being tried in certain first line indications, but I assume that's just a matter of a couple of years before we might be able to see that in first line in a more categorical sense.
Thank you. How vertically integrated do you intend to be in manufacturing over time, and what does that mean for capacity intensity?
Sure. So we actually find that needing to invest an enormous amount in CapEx, especially as a small and early company, is a very dangerous thing to do because if you don't have the utilization, obviously, then it becomes this massive millstone around your neck. So we work with CDMOs on a contract basis. What's very important to note is we're able now, one donor can give us 100 patient doses, and we can cycle 100 donors through a very small footprint, clean room footprint of about 15,000 square feet per year. So we're already up to 10,000 potential doses per year without even breaking a sweat and without investing any additional finances. It kind of became, I think, a little bit trendy about 8 years ago for a lot of these initial startup companies when money was a lot easier to build big manufacturing plants. We don't currently intend to do that. We don't see a need to do that. All of those companies, even the Gileads, Novartises, and BMSs of the world, are struggling to handle 4,000 autologous patients per year. And we can do 10,000 again with a fraction of the footprint at a fraction of the cost.
Thank you. And what do you anticipate the commercial adoption curve to look like, particularly versus established autologous CAR T products?
Yeah, well, so we talk to key opinion leaders all the time, the physicians that are on the front line and that are doing this. And what we know is the minute an allogeneic CAR T is able to show efficacy and safety that's comparable to an autologous will dry up very fast. And we would expect that to happen within a year or two. It's just autologous is such a laborious, difficult process, and the patient has to wait generally 3 to 4 weeks to get the material back, during which point they may deteriorate further. So every physician that we've ever spoken to would prefer to use an allo every day of the week and twice on Sunday if they could. The problem has just always been that there aren't any allos to do that. And a lot of people have kind of given up hope that there ever would be an allo to do so. So, the boy has cried wolf many times. And we understand that there's some skepticism in the field right now because of all the previous attempts. All we can do is just generate the data. But if the data is what we think it is, that adoption curve will be extraordinarily fast. I would say I would give it 1 to 2 years. And in most scenarios where we're able to show head-to-head kind of comparability, the allo will get adopted.
Thank you. The Cleveland Clinic just announced a 68-year patent with refractory multiple myeloma reached a 5-year remission point after an allogeneic CAR T. How does that treatment differ from yours?
Yeah, maybe I hesitate to turn it over to Reynold here because I know, Reynold, your voice is cracking a little bit, but I don't know if you wanted to weigh in on this one or not.
Sure. My apologies. Just have a little bit of a cold here. It's a little difficulty speaking. That patient in reference there is been treated with a product from Allogene. That product is no longer being developed. So it was successful in that patient, which is amazing for that patient in terms of giving them a durable response. But overall, that program was not successful. Furthermore, I see there's also another question around Allogene in general. So maybe I'll just take that since we're on the same topic. Allogene is a company that was formed around the use of knocking out CD52, which is a gene targeted by a drug called Campath or alemtuzumab. Ultimately, that it does not appear to have been successful. They've stopped pursuing that particular approach. It basically would cause long-term lymphodepletion in the patients, which would prevent the patient's immune system from rejecting the allogeneic CAR T. But as I mentioned, due to a variety of toxicities and other issues, that overall approach is not being pursued anymore.
Okay, thank you very much. And what makes allo CAR Ts so much cheaper to manufacture than auto ones?
Yeah, they're actually not too much cheaper to manufacture on a case-by-case basis. The difference is that we can manufacture 100 doses from a single donor with the allo, and when it's autologous, we have to. There's only one batch. It's the patient's batch, and that goes to the patient. So there's 100 times leverage, theoretically 100 times leverage. It turns out that the economics don't quite achieve that, but we can achieve a 20 times cost efficiency with the allo process.
And your switchable CAR T system introduces an additional control layer. How important is that on-off functionality from a safety, regulatory, and commercial standpoint?
Yeah, so the switchable CAR that I demonstrated, that's AVC201. The reason why we use a switchable in that case, but we don't always use it, is we use it when the target on the cancer cell is potentially dangerous. And by that I mean there are some targets that you identify that are great for targeting cancer, but they naturally occur on healthy cells. So your CAR Ts can have what's called an off-tumor on-target effect. And that introduces toxicity. They're killing healthy cells that they shouldn't be. That may manifest in terms of a rash or a variety of other things. So with AML, one of the reasons why it's been so tricky is because there are no really good clean receptors that only occur on AML cells. CD123 is the receptor we go after. It does occur on healthy cells. We want to be able to turn the CAR on and off very quickly. And indeed, between this and a previous trial that we did, we had 44 total patients, of which 8 of them did have some toxicity emerging, but we were able to turn the CAR off simply by stopping the infusion of that linker molecule. So that's where it's important. It's a hammer. Not everything is a nail. With things like CD19 and B-cell lymphoma, we don't need to use that because we can control the toxicity, and there's lots of experience doing so. But with AML, it does turn out to be important, and we do believe, I didn't talk about future opportunity because there's all kinds of other tumors that we can go after. It's believed that solid tumors, this issue of on-target off-tumor will be much more, prevalent in solid tumors. So having something a switchable that works as well as a conventional CAR is a big bonus and will help us with other future indications, particularly in solid tumors.
And how large then is the TAM for your lead and follow-up programs if your platform performs as expected?
Yeah, so the total potential market here would be somewhere around the $30 billion mark for a combination of B-cell lymphoma, multiple myeloma, and AML. I'll just group those are kind of the big three of the targets. We have the active clinical programs in both AML and B-cell lymphoma. We have the potential to go into multiple myeloma very quickly. We haven't done that just because of bandwidth, but we're talking about, a $30 billion market quite easily, that's currently underserved by about a factor of 5.
And so, if understood correctly, you have to infuse a bispecific antibody in order to target your allo CAR T. Is this an approved bispecific? Which one, and how does this increase the cost of the treatment?
Yeah, well, the bispecific is very, no pun intended, specific to this particular thing. So we make it. It's not sort of like a repurposed bispecific from some other indication. And that's because our universal receptor on that CAR is very unique. It's physiologically inert. That guarantees that the CAR doesn't do anything unless it's in the presence of the other bispecific. So we do make it. It's unique to us. As I mentioned before, we're only using it in those cases where the potential target could be dangerous. So we don't use it in AVC 203. We're only currently using it in 201. The cost is comparatively minor. If you make that at scale, it's the typical cost of making antibodies. We've estimated it. It's in the $500 to $2,000 range per patient estimated right now. So it doesn't really move the needle in terms of cost of goods. Again, we're talking about, the car itself we make for well under $10,000 per patient. bispecific may add about a $2,000 to that. And then, the current market price for CAR Ts is in the $350,000 to $450,000 range. So margins should be well above 90%, even if we decided to, discount the price of the allo to make it more accessible.
And in the second program, you were targeting CD19, CD20. Can you speak to the importance of targeting both receptors versus one?
Sure. Yeah. So there's been some really interesting, I mean, it's a great question. There's been some really interesting clinical data that's just been posted over the course of the past 6 months by three other companies that all have autologous CD1920s. These are J&J that bought their compound from a Chinese company called Able Zeta for 250 million upfront. There's Kite Gilead, who have their own homegrown CD1920, and then there's another company called Lyell, who bought theirs through a company called Impact. So three companies, three candidates. All three of them are now posting data in B-cell lymphoma, and all three of them are hitting complete response rates a full 20 percentage points higher than the existing CD19 only marketed CARs. So that's moving the needle from about a 60% average CR rate to about 80%, which is, very significant. And it appears, even though all the data is not in, that the relapse-free rate is, again, also, commensurately higher. So we know that CD19 plus 20 is significantly more efficacious. There are other candidates out there that show a precedent for that. The reason is because there's multiple reasons. Part of it is because when you bind both things at the same time, you can create a more effective synapse. There's more avidity and involved with that binding. The other is a lot of patients will actually lose the expression of CD19 when CD19 is being targeted. The cell kind of works its way around that. So if you have CD20, you basically have relapse insurance. So if the patient loses CD19 and they're still expressing CD19, you can still get those cancer cells.
Thank you. What are the key clinical or operational risks that could meaningfully alter timelines or valuation, and how are you mitigating them today?
Yeah, I mean, great question. It's something that, every biotech has to do. We're in the clinic and if we did an S1, you would see 25 pages of, the typical risks that we encounter. The ones that are more germane for us, we have to be very careful. We're in a trial. AML is a disease where the patient is extremely sick, and our patients typically have a 1 to 2 month lifespan when they come into the trial. We have not had any patient deaths on trial yet, but patient deaths and other kind of severe adverse events are things that we need to be very careful about and worry about. Again, having that switchable system for AML has allowed us to, I think, avoid any real serious adverse events. We've had no ICU admissions, no deaths on trial. But that's a significant one, I would say. We have, manufacturing is always a risk, but again, moving from autologous to allogeneic is a huge sigh of relief because we can plan for that well ahead of time, and we do have a GMP process, and we've created material. I would say, there is a current environment with the US FDA that is a little bit, uncertain in certain respects. They were, I have to say, they were amazing at getting our IND approved, well, right at the 30-day deadline. So it hasn't impacted us yet. But those are the kinds of things that can always arise and put you off track a little bit or introduce delays.
And keeping on the FDA, the bispecific will have to be approved separately from the allo
Yeah.
CAR. And how do you get that through the FDA?
Yeah, so they are considered inert, each one by themselves. So they're non-active if they're given separately. So they're not they're approved as a pair. It's all approved as the same under the same IND as a compound kind of medicine, but they don't have to be approved separately. No.
Okay. And last question, what inflection point in the company will the current raise allow you to achieve, and what will be the use of your proceeds?
Sure. Of course. So, in terms of the use of proceeds, they are directed, we are very streamlined and extraordinarily cost conscious. They will be directed exclusively at our two active clinical programs, AVC 201 and 203. Our expectation in 2026, which is, sort of the majority of this timeline, is that AVC 201 for AML will complete dose escalation. And again, that's just 3 or 4 more patients into the first quarter of 2026. And then, if everything looks good, we'll embark on dose expansion. So that'll be a very significant dose expansion should be able to be completed within 2026. That'll be a very important body of data from 20 patients that basically demonstrates whether we believe that this is a drug or not at the given dose. So that's a very important one. And again, that's the kind of thing, the body of data that generally would also inspire a strategic to generate a business development deal. So that's what's happening with 201. And with 203, the most important value inflection point will be our initial 12 patients in dose escalation. We just have two cohorts in dose escalation. Both of them we believe will be clinically meaningful. So by middle of the year 2026, we will be able to generate data that will essentially kind of definitively state that we have an allo that works as well or better than an autologous, and whether or not there's permission to believe that our AVC 203 can be as good or better than existing marketed CAR Ts. And that'll be, obviously very significant. If it's as good or better with a price that's 20 times cheaper to make, it'll, it's kind of a no-brainer that'll be something that everybody's going to want.
Great. Well, thank you very much. And thank you to the audience for the great questions. If you have additional questions or want to be connected to AvenCell, please reach out to me. Everyone has my email address. I'm happy to facilitate that conversation. In turn, they will have all of your information as well and will be reaching out. Andrew, I want to turn it back over to you for any final words before we close out today.
Just again, a big thanks to Force Family Office for what you guys are doing and the networking that you're doing. We're really excited to connect with family offices who are, very inspired by the same mission that we are. We're very transparent, happy to answer any questions. We like to think that we're pretty good at getting back and responding to inquiries and so forth as well. So for those of you on the call who are interested by what we're doing, please do reach out through Callie and the team. We'd love to hear from you. Thanks so much for your time and attention today.
Thank you, everyone, and have a great day.